in Dr. Darren Rokita's lab at Florida State University, and I'm going to tell you guys a little bit about my dissertation project today. So the overall goal of my research is to use experimental evolution to identify genes involved in venom resistance. And this has a wide range of applications, from ecology and evolution of predators and their prey, as well as the cellular and molecular mechanisms of venom toxins, and in therapeutic development. Of course, the larger goal of my research is made up of a lot of smaller experiments, and so today I'm going to try to convince you that the time I've spent injecting fruit flies with centipede venom has not been a complete waste of time. So, to backtrack a little bit, I'm sure many of you have heard this spiel before, but venom is a complex cocktail of toxic proteins and peptides, and it's produced by a variety of species across the animal kingdom, from invertebrates such as centipedes and scorpions, to vertebrates such as reptiles, and even in some mammals. It's primarily used for predation and defense, and though the complexity and relative amounts of certain toxins differ from species to species, there's a high degree of homology between these species in terms of the toxins that are available and their function. Okay. And so the evolution of venom is well accepted to be this co-evolutionary arms race between predator and prey. Whereas the prey becomes more resistant to the venom has to become more potent or efficient to combat that resistance. And just here's one example of that. It's in the eastern diamondback rattlesnake and the cotton rat. And venom is also used in medicine, not only in the creation of antivenoms, but also in treatment of human disease. So it's used in treatment of diabetes, heart and blood disease, and pain management, and it's also being investigated as a possible cancer treatment. But many of these venom-derived medicines come from well-characterized venoms that are based on trial and error methods that largely involve interaction of just protein-protein interactions and also protein-small molecule interactions of an individual toxin. So these don't actually encompass a whole venom or a whole animal system, and they also largely ignore a large group of venomous animals, such as the invertebrates. So this leaves a lot of the venom toxins and their targets actually undiscovered. So what I'm hoping to do is design this experiment to bring molecular coevolution and therapeutic discovery together, employing the powers of natural selection in experimental prey populations. And with that experiment, I'm hoping to turn this into something more like this and address questions surrounding venomous predators and their prey and those coevolutionary dynamics, look at the identified targets of venom toxins, and lead to more efficient medical uses. So the overall design of my experiment is an evolve and resequencing study using Drosophila melanogaster and venom from the Florida blue centipede, Scolopendra viridis. And the overall design of my experiment is basically that I inject these flies with very dilute doses of venom. The survivors are then selected to parent the next generation, and this process continues generation after generation until eventually all of the venom-injected flies begin to survive. Of course, the reality is a little bit more complicated than that, so I'll just walk you through step by step what a general evolve and resequencing study is like. So here you see that there are initial inbred lines that are mixed neutrally for several generations to create a genetically diverse population. From here, individuals with an extreme value of a given trait are selected, and this selection continues for several generations until eventually the ending populations are sequenced and compared for allele frequency differences. In the case of my experiment, you can assume that this blue side here are those that survive venom injection, and this red side over here are those that die. Of course, we can't really select for death, so you can pretty much ignore this side of the equation, but I do want to point out that I will be comparing this surviving group to a control group that's injected with sterile phosphate buffer saline, all other experimental conditions identical. So why am I using Drosophila? It should be somewhat obvious for those of you who have worked with Drosophila, but they are a model organism in genetics research. They have an extremely well-studied genome with about 75% homology to human disease genes. And most importantly, venom by definition must be injected to observe a physiological effect. And so they offer a whole animal invertebrate system for which to observe these effects. So in order, though, to justify this evolving resequencing study, because this is an experimental or novel approach, we have to first establish that there's enough variation in these inbred lines that this trait can be selected for and evolution can be observed. And so that brings me to my main question of this talk is, can this novel system be used for an experimental evolution study? So to start to get at this, after obviously extracting the venom from the centipede, I had to figure out what dose was appropriate to inject into these flies. And so to do that, I performed the LD50 assay, which is a lethal dose 50, which is the dose that causes 50% mortality in the population. And I performed this assay on 5 different inbred lines with 3 replicates of each line, and ultimately found the median LD50 of 1.09 nanograms per milligram. I then took this median dose and injected it into an additional 53 inbred Drosophila lines to look at variation in this trait. And for that assay, I used 25 males and 25 females plus a control group. And then I used an exact binomial test to come up with a death probability for each of the lines. And here are the results of that study. So you can see we have quite a lot of variation. So again, over here on the y-axis, I have the death probability. And down here, I have all of my fly lines. This black line in the middle represents the null hypothesis of a 50% death probability, with the dotted lines being the 95% confidence interval. So the lines in blue are significantly differ from the null hypothesis in that they're more likely to die. Those in greens significantly differ from the null hypothesis in that they're less likely to die. And these here in red meet that null hypothesis that we expect about a 50-50 death probability. Okay. But I wanted to make sure that this variation was actually due to the venom itself instead of another factor. And so we know that male Drosophila are about 15% smaller than the females, and because they were all injected with the exact same dose based on an average fly weight of 1 milligram, I wanted to see, was there a difference in sex between the flies? And I expected to see for there to be a difference, right, as the males are quite a bit smaller. So I ran an ANOVA, and I did find that yes, males are much more likely to die by about twice as much. But I still wanted to know, is this actually due to weight or is it due to sex? And so my next question is the fly weight correlated with death probability? So taking the actual average fly weights for each line, I tested for this and ran a linear regression analysis and found that it's actually not correlated. So here you can see on the y-axis I have my death probability, on the x-axis I have my average fly line weight distributions, and you can see with the same coloration, so the blue lines here that are more likely to die have a range of weights, as well as the lines shown in green that are less likely to die have a range of weight distribution. I performed the same analysis within the sexes, and we see similar patterns between the females and the males. And so, yes, there is a significant difference between the males and the females in response to venom injection, but this difference in response is independent of size. So what this tells us is that this system can actually be used for an evolving resequencing study and help us address questions not only in identifying the targets of venom toxins, but also in some of these complex coevolutionary dynamics between venomous predators and their prey. So can this system be used as for an experimental evolution study? Yes, these preliminary results point to that it can be. But I was able to go one step further because I used the flies provided from the Drosophila synthetic population resource, which if you haven't heard of them, I highly recommend that you look them up. Here's the website here. And they offer a huge range of resources that enable you to perform quantitative trait loci mapping very easily, including even an R package that pretty much does all of the work for you. It's fantastic. So I actually performed this mapping in my small data set, or I would rather just call it mini mapping, if you will. And I got these results. So this line here represents the kind of default significance threshold for the quantitative traits. And so as you can see, I did find one QTL that met that significance threshold. And within that QTL, I found 20 genes that I was able to use fly-based to actually pinpoint the exact location. It's about an 80,000 base pair range. And with these 20 genes, very few of them had enough information for me to really get at what their functions were. But I was able to proportionally group them. And so most of them were involved in general protein binding. And there's also a handful of them that code for proteins that have zinc ion binding domains, which are actually thought to be applicable for host defense. So, in summary, yes, we established variation in response to venom injection in Drosophila. There is a sex difference in response to this venom injection, but this difference or this variation is not due to size. And so yes, this system can be used for an experimental evolution study. We were also able to identify a significant QTL containing 20 genes that may be involved in venom resistance. And so we will be able to use this experimental evolution approach to address coevolution and identify targets of venom toxins. And so the next steps are obviously to go ahead and move forward with this evolve and resequencing study. And the reason why I wanted to do an evolve and resequencing study is not only to identify these genes responsible, but also to look at some of the life history traits that might be changing over time as a result of this injection, to see if there are any fitness trade-offs as a result of developing this resistance. And so with that, I guess it went relatively fast. So here are my acknowledgements. Like I said, I'm in the Rokita lab. I've got a huge number of undergrads, more actually than are listed here now. I also get a substantial amount of help from Kim Hughes and her lab, and the DSPR funding, NSF, FSU planning grant. And I also want to plug, since I talked about some cool things that I wasn't able to show here today, we have a Facebook page where you can see a bunch of videos of venom injections, extracting venom from centipedes and scorpions and snakes and all that fun stuff. So I will take any questions.
I have a two-part question. How long do these flies live? And then is there some idea to potentially investigate a preference for age independent of size?
So that is a good question. Flies can live for a decent amount of time, but usually their life cycle is about 2 weeks. But I should note that to control for age, I actually inject all of the flies within 1 to 3 days of them emerging from the pupa. And I also inject them between 1 and 3 hours of dawn for the flies. So they're all injected at about the same age and the same time of day, because throughout the day their metabolism can change. And so I had to control for those factors.
I guess my question was if there might be a preference for age, like because you're talking about the preference for that the males are more likely to die independent of size.
I would assume, I haven't looked at that because they're all the same age really, but I would assume that those that are older are going to be more likely to die. Does that sort of answer your question? Okay.
I'm not misunderstood, but are you saying that you're not going to do a susceptibility line? Like a line with increased susceptibility because they're going to die?
So I'm planning on using so once I have my established lines for my actual evolve and resequencing study, which is actually going to be a genetically mixed population, so I'm going to have to recalculate an LD50 for that population, and I'm going to use that same dose throughout the experiment. So eventually they'll start to die less.
Yeah, we'll talk.
Okay. Yeah, sure. Thank you.